Hello students, welcome to EPG Pathshala. I am Dr. Soma M. Ghorai from Hindu College, University of Delhi. Today, we are going to discuss about the module Major Histocompatibility Complex from the paper Immunology. After studying this module, you shall be able to recognize the various haplotypes of the major histocompatibility complex and their different structures. You will be able to differentiate between class 1 MHC and class 2 MHC antigen presentations. You learn more about the role of MHC in self and non-self recognition, and you are able to ascertain about the genetic architecture of the major histocompatibility complex. Learn more about the MHC polymorphism and their basis on natural selection process. How epigenetics has a role in MHC polymorphism. You know more about the role of MHC in autoimmunity and other diseased conditions, and why the MHC, which is known as human leukocyte antigen genes in humans, is useful for typing and is used in transplantation clinics. For the body to rein an attack on any pathogen, it must be recognized as self or non-self, and this is accomplished by a set of cell surface receptors which not only distinguishes self from non-self but also plays a pivotal role in tissue transplantations and rejection, autoimmunity and susceptibility to infections. These cell surface receptors are encoded by most diverse gene loci in vertebrates known as major histocompatibility complex class 1 and class 2 loci. In humans, the major histocompatibility complex spans a considerable part of the DNA, that is about 0.1% of the total genome contains more than 200 coding loci for these genes. Thereby, they are highly polymorphic genes that can be attributed to the natural selection over long periods of the evolutionary time that we have scaled. Other than any other regions of the genome, the MHC genes are the most associated with the autoimmune and infectious conditions. These disease-associated variations in the major histocompatibility regions are mainly due to very subtle and minute changes in the gene loci. But there is still more to learn about the MHCs. Like, we should know what are the mechanisms that drives this MHC polymorphism, which are the naturally and positively selected variations having functional importance, and why the signaling pathways does the MHC genes follow at the molecular level. Zinkernakel and Doherty in 1975 found that T cell responses were restricted to antigens only when they are presented along with the major histocompatibility complexes. In 1980s and 1990s, we finally revealed the structure of MHC molecules and their encoded proteins. Role of MHC in self and non-self recognition. T cells cannot distinguish between self and non-self peptides. During the early development, most of the immature and undifferentiated T cells from bone marrow that encounter self-antigens are eliminated. The maturation and proliferation of T cells happen in the thymus via random gene rearrangements of the TCR genes. This random generation of huge repertoire of T cells also generates 95% of those cells that bind to self-antigens and are eliminated. The only T cells that mature and enter the circulations are those that recognize the peptide MHC complex. Thus, though thymic selection, MHC genes influence the development of T cell repertoire. Now we will be talking about the MHC haplotypes. The MHC class 1 is the first haplotype. These are the heavy chains encoded by 3 genes, HLA-A, HLA-B and HLA-C and presents small peptides to cytotoxic T lymphocytes. Almost every nucleated cells of the body present MHC-I molecules. Mostly, these peptides are self-peptides, but in case of virus-infected cells or cancer cells or even parasitic infected cells, the foreign peptides are presented, resulting in the elimination by the CTLs itself. The CTLs are very unique cells with highly specific T cell receptors that scan the MHC antigens complexes and CT8 positive molecules to stabilize and help bind these cells to the MHC antigens. Now we are talking about the structure of MHC-I. In this figure, we see that MHC class I is composed of alpha and beta polypeptide chains held together with non-covalent bonds with a short cytoplasmic tail. The alpha chain, which is about 45,000 daltons, is the MHC encoded integral membrane glycoprotein, while beta-2 microglobulin, which is about 12 kilodaltons and is non-MHC encoded extracellular peptide. The alpha chain is of 350 amino acids with 3 globular domains, alpha 1, alpha 2 and alpha 3. It also has a 26 hydrophobic amino acids transmembrane segment spanning the plasma membrane. The alpha 1 and alpha 2 forms the platform as the peptide binding unit, mainly comprising of the beta sheets. The groove formed between them is blocked at both the ends and is enough to hold about 8 to 10 amino acids. The beta-2 microglobulin chain is non-polymorphic and can hold about 90 amino acids, peptides, and is encoded by chromosome 15 and interacts non-covalently with alpha 3. This part of the MHC molecule binds to the T cell cytotoxic. Class II MHC molecules. Now the class II peptides are expressed on specialized cells called antigen presenting cells, mainly the macrophages, the dendritic cells and the B lymphocytes. The class II molecules are encoded by 3 polymorphic genes of the MHCs, the HLA-DR, the HLA-DQ and HLA-DP that binds to different peptides. The immunodominant epitopes are largely confined to the pathways dependent on lysosomal degradation and peptide binding of the newly synthesized MHC-II molecules in the acidic endosomal compartment that can be either in the endoplasmic reticulum or in the Golgi or even in the lysosomes. The MHC molecules that are involved in the CD4 positive T cell responses against the intracellular pathogens may trigger immunity towards infection, thymic selection, autoimmunity as well as tumor immunity. Here we are talking about the structure of MHC-II. Like MHC-I, the class II MHC molecules have an extracellular domain, a transmembrane domain and an intracellular carboxy-terminal tail. The extracellular domain is composed of 2 non-covalently associated domains, alpha 1, alpha 2 or beta 1 and beta 2 of about 90 amino acids which are anchored to the cell membrane. The heavy chain, which comprises of alpha 1 and alpha 2, has a molecular weight of about 30 to 34 kilodaltons, and the light chain, beta 1 and beta 2, has a molecular weight of about 26 to 29 kilodaltons. The transmembrane domain is comprised of about 25 hydrophobic amino acids, and the cytoplasmic tail is of about 10 to 15 hydrophilic amino acids. 13 to 18 amino acid residues of antigenic peptide binds to this region of the antigenic peptide binding domain. In the figure A, it shows the peptide in MHC class 1 binding groove, and in the B, it shows the peptide where MHC class 2 binding groove. These peptide binding grooves of MHC-I and MHC-II proteins shows that the groove are usually of 8 to 10 amino acids long. The residues at both the N-terminus and C-terminus end of the peptides are anchored in the groove, and it forms a peptide backbone which is usually above the binding groove. In the B, peptide binding in an MHC class 2 groove usually contains 13 to 18 amino acids long peptides. These residues bind at both the ends as well as in the middle, so it do not form any of the raised area above the binding groove. The sources mainly are by the major histocompatibility complex in the immune responses. Here we see the three-dimensional figure of class 1 and class 2 MHC binding peptide grooves. The cleft of MHC-I and class 2 molecules fold into a highly similar conformations featuring a peptide binding groove to present T cell epitopes. Binding of the peptides to the grooves of the MHC-I molecules are composed of 2 alpha helices and 8 beta strands formed by 1 heavy chain, while MHC-II uses 2 domains from different chains to construct the peptide binding groove. The peptides bind to MHC molecules through primary and secondary anchor residues protruding into the pockets in a peptide binding grooves. Peptide preferences dependent on the amino acids polymorphism comprising the anchor pockets, which are related to the various alleles of MHC. The conformations of peptides presented by class 1 MHC molecules are length dependent. The modified groups of post-translationally modified peptides have important roles in the peptide MHC interactions. Talking about class 3 MHC molecules, now these molecules are mainly comprised of serum proteases and complement enzymes and do not have any role in antigen presentation. The complement proteins or the component includes the C2, the serine proteases, the C4a and C4b, which are the pro-proteins of the multi-chain forms, and factor B, and this is the component of the alternate complement pathway. They also include the tumor necrosis factor alpha and beta as well as certain heat shock proteins. Here I am presenting the MHC class 1, class 2 and cross presentations of both of them. This figure represents antigen processing and presentation pathways in the dendritic cells. MHC-I pathway, where intrinsic pathogens like viruses or endogenous proteins are either degraded in the cytosol or nucleus by immunoproteasomes and are presented as peptides along with MHC-I to CD8 positive TCTL cells. Whereas in the MHC-II pathways, we see the exogenous peptides are endocytosed and via endomembrane system bind to the MHC-II to be presented to the CD4 positive TH cells. The cross-presentation pathway indicates that the exogenous peptides which has been sequestered via endocytic pathways or mechanisms may compete for the peptide either to MHC class 2 or MHC class 1 pathway. Some dendritic cells have a unique ability to deliver the exogenous antigens to the MHC class 1 pathway. Now this figures represents the class 1 molecule pathway and the endogenous proteins which are broken down by proteasomes into smaller peptides. Here we see that in the endoplasmic reticulum, the antigenic peptide binds to the peptide binding site in an MHC class 1 molecule. The peptide MHC complex then migrates through the Golgi apparatus via the endomembrane system to the cell surfaces, or we can say the plasma membrane of these cells. This slide shows the class 2 MHC molecule pathway, where the alpha and the beta subunits of the class 2 molecule bind to an invariant chain that is represented as big I and a small I, and that happens in the endoplasmic reticulum. The invariant chain is partially degraded in an endosomal compartment. The portion of the invariant chain that occupies the antigenic peptide binding site of the MHC class 2 molecule, or also called as CLIP, C-L-I-P, is removed with the help of HLA-DM, freeing the molecule for binding to other processed antigen. Once the antigenic peptides are bound to an MHC class 2 molecule, then the complex migrates to the cell surface or the plasma membrane. When we talk about the genetic architecture of major histocompatibility genes, now the MHC genes are located in the autosomes, the Y chromosomes, and even in the mitochondrial genome. It is a large chromosomal region of chromosome 6 that has over 200 coding regions, and apart from specific immune functions, these genes also impact growth, development, reproduction, odor, and olfaction. It is extremely difficult to unravel the functional aspect of each loci owing to its high gene density, extreme polymorphism, strong linkage disequilibrium, and clustering of the genes with related functions. Hence, our understanding of MHC-related diseases are very poor because, number one, the natural selection should have eliminated the neutral and deleterious alleles and must have kept only the disease-protective alleles. Second, they might have used the specific variations among the sea of various MHC gene variations which are functionally relevant and can be kept. Third, the MHC gene-related diseases may not be in the genes, but they can be due to certain discrete changes in the amino acid or in the changes in the groove residues. This slide shows the genetic architecture of the major histocompatibility complex in humans as well as mice. Here you can easily see that the MHC complex is situated in chromosome number 6 of humans and chromosome number 17 of the mouse. There is stark differences in the locations of these MHC molecules. In humans, MHC-II is juxtaposed within MHC-I and MHC-III, whereas in case of mouse, MHC-I, II, III are lined, and later on the polymorphic form of class 1 MHC is seen in details. The MHC in mice is termed as H2 or histocompatible 2 complex, and in humans it is known as HLA or human leukocyte antigen complex. The immune responses specific regions for MHC gene cluster, which are known as classical, are closely located linked genes and are called class 1 and class 2 loci and are most divergent among vertebrates. Among the most studied, humans have 6 class 1 loci, class A, B and C, and 8 class 2 loci, DP, DQ and DR. Whereas in mice, there are just 3 class 1 loci, namely K, D and L, and 2 class 2 loci, A and E. In mice, the class 1 loci are bifurcated into 2 portions, one at the end and the other at the front. Whereas In humans, the chromosome number 6, we see here the class 1A and class 1B comprising of the classical and non-classical loci are present after the MHC class 3 positions. This shows the phylogenetic tree whereby we can see that all the immunoglobulin are formed from a common ancestors. The immunoglobulin multi-gene superfamily gene basically gave rise to not only the immunoglobulins but also the T cell receptors, various antibodies, as well as the major histocompatibility genes, suggesting that all of the molecules evolved from a common ancestor. The polymorphism can be observed with over 170 to 100 alleles per loci, with each locus having multiple coding genes. The close linkages among all these loci means that each individual inherits a particular combination of major histocompatibility complex alleles as a single haplotype. Thereby, there are no or little chances of recombination, separating these loci, thereby resulting in highly polymorphic status. Here depicts the chromosome number 6 specifically in the humans, where we can see the chromosomal location of all the MHC locus depicting highly polymorphic tendencies. All the alleles of the MHC molecules show polymorphism and can be observed over 170 to 100 alleles per locus, with each of them having multiple coding genes. We can see here the close linkages among these loci, and that means all of them have to be inherited together. So, it shows just one single haplotype. Thereby, no combination or little combination can be depicted within themselves. Thereby, we can see that all the MHC polymorphic genes are inherited in totality, resulting in high polymorphic status. We talk about the MHC genes polymorphism and how they are taken up by the simple natural selection processes. Now, there are many evidences that confirm that MHC polymorphism is maintained due to natural selection, like high number of alleles, uniform allelic frequencies, deficiencies in homozygotes, linkage disequilibrium among the various loci, high DN upon DS substitution. Now, DN upon DS means the non-synonymous and synonymous substitutions of various amino acids that has taken place within the gene loci, the ancient allelic linkages, and disassorted mating preferences. MHC polymorphisms, though, is an important for disease resistances. It is also involved in promoting outbreeding. MHC polymorphism is basically aimed to provide herd immunity. What is herd immunity? Which is basically a large number of variables are present in a population, then it is more likely that one particular infection may not get an escaped route and spread is restricted, thereby making that population safe from any of the diseases that has come and infected it. Thus, it can be related to the potential of certain populations to their relationship with the pathogen-associated selection. In human evolution, certain haplotypes of MHC were introgressed into the Eurasian and Oceanian populations much before they were introduced into the African population. The polymorphism among the MHCs has been useful in tracing the ancient population migration. For example, now this can help deduce the divergence of early humans from their African origins and can be explained on the basis of admixture with the archaic humans. The epigenetics and the MHCs, they are both closely linked. At times, it is difficult to explain why certain phenomena through conventional genetics cannot be solved, and there are certain such cases where expression of MHC genes was shown to have an epigenetic effect. The famous example being that of the offsprings of mothers which carry the alleles for MHC-II, especially the HLA DR B1*15 for multiple sclerosis. Now, this is an autoimmune disorder and are more likely to get infected rather than the fathers carrying this particular affected gene. Significant epigenetic modifications such as methylation, acetylation, and histone modifications have been demonstrated to alter the MHC class II regulator. Now, this is known as C2TA expression. Therefore, many diseases associated with aging and autoimmunity is now linked to the polymorphism of C2TA expression. The major histocompatibility complexes are very important in transplantation immunology. The allogenic immune responses. When a tissue transplant is performed, the human leukocyte antigen of the donor is recognized by the recipient immune system, triggering an allogenic immune response. Allogenic means within the same species. The divergent alloantigens that cause due to large number of genetic differences, which we can now relate to the highly polymorphic conditions of the MHC molecules between the donor as well as the recipient, are easy targets for T cell recognition, thereby increasing the risk of both graft rejection as well as graft versus host disease, known as GVHD. Here in this slide, we see the direct recognition of both the CD8 positive TC cells and CD8 positive T helper cells of an intact major histocompatibility molecule displayed by the donor antigen presenting cells in the transplanted graft, as well as we also see the indirect recognition pathway whereby the donor's major histocompatibility complex with the foreign peptide is processed and presented by the recipient antigen presenting cells, which can either be the dendritic cells or simply the macrophages, and are presented to only the C4 positive T helper cells, causing the delayed type hypersensitivity reaction. Traditionally, all the HLA typing was performed by serologic testing using antiserum in complement-dependent cytotoxic assays. But recently, more precise DNA-based human leukocyte antigen typing methods are being used, namely the sequence-specific oligonucleotide probe hybridization, the sequence-specific primer amplification, the sequencing-based typing, and the reference strand-based conformation analysis, which are being frequently used these days. And HSCT donor is referred to as a 10/10 allelic match or a perfect match, when both the HLA alleles are identical at each of the HLA-A, HLA-B, HLA-C, HLA-DRB1 and HLA-DQB1 loci in case of both the donor as well as the recipient. While searching for an unrelated donor, high resolution should be about 4-digit genetic typing of both the patients and the donor is necessary. Otherwise, we are now sure that will lead to transplantation immunology and rejection of the grafted tissue. So students, let us summarize what we have learned in this module. The major histocompatibility complex. Now we know that it is known as MHC class 1 and class 2 loci are cell surface receptors encoded by the most diverse gene loci in vertebrates and spans about 0.1% of the total genome, containing about 200 coding loci. The MHC genes are located in autosomes, Y chromosomes, and even in mitochondrial genome. They are basically located in chromosome 6 of humans and chromosome 17 of mouse and has 200 coding regions. The MHC in mice is termed as H2 or histocompatible 2 complexes, and in humans it is known as human leukocyte antigen complexes. MHC not only distinguishes self from non-self but also plays a pivotal role in tissue transplantation and rejection, autoimmunity, and susceptibility to infections. Apart from specific immune functions, these genes are also impact growth, development, reproduction, odor, and olfaction. Class 1 heavy chains are encoded by 3 genes, HLA-A, HLA-B, and HLA-C, and presents small peptides on the cytotoxic T lymphocytes, enabling each cell of the body of the immune system with possible chances of infection. Therefore, every nucleated cells of the body present MHC-I molecules. Class 2 peptides are expressed on specialized cells called antigen-presenting cells. They are mainly the macrophages, dendritic cells, and B lymphocytes. Now, these molecules are encoded by 3 very small polymorphic genes, the HLA-DR, the HLA-DQ, and HLA-DP that bind to different peptides. Both MHC-I and MHC-II presents peptides to the CD8 positive and CD4 positive T helper cells. The sources of these peptides may differ. The MHC-I are presented with intracellular peptides and exogenous peptides are represented by the MHC-II. Both MHC classes have similar three-dimensional structures. They originate from the common parent gene by gene duplications and a similar function in the antigen presentations to the T lymphocytes. MHC genes influence the development of the T cell receptor through thymic selections, as only those T cells that recognize the peptide MHC complex are able to mature and enter the circulation. There are many evidences that confirm that MHC polymorphism is maintained due to natural selection, like high order number of alleles, the uniform allelic frequencies, the deficiencies of homozygotes, the linkage disequilibrium among the loci, high DS and DN substitution, and ancient allelic linkages, as well as disassortative mative preferences. Expression of various MHC genes was shown to have an epigenetic effect, as many diseases associated with aging and autoimmunity is linked to the polymorphism of MHC class II transactivator. This is an enzyme. The allogenic immune responses happen due to divergent MHC alloantigens between the donor and the recipients, which are easy targets for T cell recognition, thereby increasing the risk of both graft rejections and also graft versus host disease known as GVHD. Precise DNA-based HLA typing methods using molecular techniques like sequence-specific oligonucleotide probe hybridization, sequence-specific primer amplifications, sequencing-based typing, and reference strand-based confirmation analysis are frequently used for HLA typing in clinics. Thank you.